Are we looking for in urease? What is the whole point is identification. What does a positive look like? Just remember? This one's the blue one. Pink. So yellow was negative. And when we break down urea with the urease enzyme, then we get what? Ammonia, which is alkaline. It's high pH. And so then it will turn pink. So this is what organism that we talked about, like that people get. If people have this infection and they do the breath test, and the breath test turns out, H. pylori. Awesome. Yeah, so that's something that we use to test H. pylori. Is urease positive? Okay, acid fast. Here we go. We can do this one. Everybody. What are the two organisms that we did for acid fast? It's okay if you can't remember. Smegmatis. And then what was our second organism that we used? And what is the whole point of the acid fasting?
To find if it has that mycelium
Yes.
wax on the surface.
I'm sorry, can you explain the mycolic wax?
Yes. So mycolic wax, so if you say mycobacterium, it has this mycolic waxy acid material on the outside. So what other organism other than Mycobacterium smegmatis, which one are we really worried about? Tuberculosis. So that's Mycobacterium tuberculosis. Tuberculosis is 2 billion people on the planet have tuberculosis. So it's a stain to test for tuberculosis that a lot of people actually do in the field, that they can do it easily in the field and test for people. We don't test it in this lab, and you wouldn't unless you were absolutely certain that you could use all your proper PPE. But you don't want tuberculosis, obviously, so we use a different Mycobacterium organism. But what color is Mycobacterium smegmatis? Pink. Because of what dye? What was the dye that we put on it? Carbonyl fusion. Carbonyl fusion is fuchsia.
Oh.
It's pink. Carbonyl fusion, fuchsia, if that helps you remember. Those are going to be bright, bright pink. Then what was our secondary stain? What was it?
I was just saying
Oh yeah. What was our secondary stain? Methylene blue. Blue. Sorry if my spelling's not right. Methylene. Yeah, that looks right. So methylene blue is going to be Staph aureus. So your Staph aureus is going to be, so if you look, if on the exam there's a microscope there and it has hot pink bacilli and blue cocci, then we know that that's probably Mycobacterium. It looks like Mycobacterium and Staph aureus. Yeah? And we're looking for tuberculosis. Okay, endospore stain, last one. Is what were our organisms?
Serious?
Yep, Bacillus cereus. And what? Trick question. Yes, Bacillus cereus, yes, good. 1 day and what?
7 days.
7 day. So what is the point of having a 1 day and a 7 day? What are we looking for?
Endospores.
Endospores. Which one's gonna have more endospores?
7-day.
Why? Yes, because they're running out of food and they're sad. So then they're gonna sporulate, they'll form endospores. What organism do we, Bacillus cereus is an organism that we worry about endospores. But what's another that we worry about that forms endospores? C. diff. C. diff forms endospores, that's why we worry about it a ton. What color are the one-dayers? Red because of what dye? What did we dye? No, this one. So remember, this is the one we boiled. We had to boil it because, so if you boil an endospore, does it kill the endospore?
No.
No, you have to autoclave it. So even though we did on Bacillus cereus, even though we did boil them to try and get the dye in there, they still weren't dead. They still don't die because they can just sporulate. So when we boiled them, what did we boil them with? Malachite green. And they're green. What about the 1 day old? Do you remember the counter stain?
Safranin.
Safranin. Okay. So that's C. diff or Bacillus cereus. Endospores. Huh.